Okay. So now let's look at this variation parts. Okay. So first thing, we do know that variations, we have two kinds of variation. We're going to look at it in this chapter. So the first kind of variations we call genetic variation. Okay, genetic variations, that from here we do know that involve 5 different mechanisms. Okay, 5 different mechanisms that can give us the genetics variations. So what are these 5? So the first, during the crossing over, meiosis. Okay, number 2, independent assortment. Number 3, okay, random mating. Number 4, then we have the random fertilizations, which we have discussed, okay, previously. And in order to create a new allele, then we have to depend on the random mutation. So these 5 mechanisms will contribute to the genetic variation. Okay, but for a person to develop, we cannot run away from another factor here, what we call it as the environmental variation. Okay, so means that, for example, identical twins, but they won't look exactly the same. Why? Because the factor from the environment, for example, diet, exposure to the sunlight. So those will give us the variations, and we're going to look at why variation is so important in terms of the selections as well as the evolutions. Okay, so that's first part. Now let us look at our learning outcomes that we need to know here. So these variations, okay, we have variation, right? Different variation. We can categorize this variation into 2 broad categories, which are continuous variation, okay, which are continuous variation compared to the discontinuous variations. Okay, we have these 2, continuous and discontinuous, and we're going to look at their genetic basis. Why these variations belongs to a continuous? Why these variations belongs to the discontinuous? Okay, so basis of continuous, we're going to look at many genes, additive gene effects. Okay, and discontinuous, we're going to look at one or few genes that control characteristics. Okay, I will go through detail later. And the second part of today's topics, we're going to look at explain by example how the environments actually will give us the effects onto the phenotypes of the plants and as well as the animal. Okay, so first thing we look at this genetic variations. So when talk about genetic variation, actually refer to what? It refers to 2 important points here, difference between the individual. For example, between you and your siblings, you and your friends. So individual. And also this genetic variation can also refer to difference between populations, between us and African, between us and Latin American, between us and, for example, Japanese. So those are 2 different populations. So we're going to look at this genetic variation, how different. Okay, so genetic variation mainly occur through the DNA mutations, or we put it just now as a random mutation. Okay, as well as the sexual reproductions, where we're going to talk about the meiosis, during meiosis, random mating, as well as the random fertilization. The last part, gene flow, here I will discuss in detail in the next 2 subtopics. I haven't talked about the gene flow, the next 3 subtopics, sorry. Okay, so this one, a quick one. Genetic variation can be introduced, okay, into population through the mutation, and this occur very spontaneously, unpredictable and spontaneous. Okay, so it's a change of nucleotides. It can be rearrangement of the gene. It can change of the chromosome structure. What we need to know here is the variations here can be introduced. For example, if let's say hair color. Okay, let's say hair color only 2, white or black. Okay, black, white and black, 2 allele. But how we get a different color, you want to increase more, right? Because no matter how you mix and match, it definitely either is black or white, for example. But now, if you want to introduce a new allele, when introduce a new allele, then definitely we have to depend on the mutation to create new allele. So when we'll talk about the mutations here, what we want to have the new allele. Okay, but generally, these new allele, they are recessive. You won't be able to see the effect until when the two descendants, they have also happened that they have recessive, then they will produce the offspring that show the recessive trait. Okay, and we do know that genetic variation also arises for sexual reproduction, involve the gametogenesis, particularly the meiosis, and also the fertilizations that what we have learned, crossing over, independent assortments, random mating, and random fertilization. Now, always remember these four only do what? They only do the reshuffling and also the recombinations of the allele. Okay? They recombine the allele, but they cannot introduce a new allele. Okay? No matter how, they just recombine the allele. Okay. So genetic variation, you can see that in the shells, okay, different color, but they belong to the same species. Okay, genetic variations, whether caused by reshuffling allele or introduced, this allele can be passed on by the parents, give a different phenotype in the offspring, okay, or the children's here. So how we divide genetic variations? We can divide into the continuous and discontinuous. Okay? These two forms, right? Continuous versus discontinuous. And now we're going to look at the basis of the genetic, how it will form discontinuous and how it form this discontinuous. Okay, so what's the definition of continuous variation? When we talk about continuous variation, basically we are looking at what? When we talk variation means different, we are looking at phenotypic difference which can be quantitative or can be measured. For example, we talk about body height and body mass. So why we use these two? Okay, we look at body height. Okay, let's say a person A, body height 180 centimeter. A person B, body height is 170 centimeter. So can you see that you show two different variations, right? Two different phenotypes. Okay, A 180, B 170. But here, if you look at the difference, we can actually measure the difference. We can quantify the measurement. Oh, their difference is. Then here, can you see that we can quantify it? So this kind of variation we term it as continuous variation. And this kind of variation we term it as continuous variations. And most important part here, you need to know this one, highlight. So if in continuous variations, what is the genetic basis here? Very, very important. Different allele at a single gene locus only give a very small effect onto the phenotype. Are you clear? Okay. So means that it will give something, but it won't give a big effect. Okay, it won't determine the overall effect. And involve many genes, a different gene, and this different gene will act together to give us the additive effect. This one we call additive effect. I'll show you later what's the additive effect. And they involve large number of genes that combine together the effect. So this one we call it polygenics. So three important points here. They involve many genes, and each of them give us additive, means that add on some effect here until we will get that variation. For example, if you look at the body height involves roughly about 180 genes that we already know. Not one gene determines your body height. Are you clear? Not one gene that determines our body height, but roughly about 180 genes involved. Each gene will they have a different allele. And this allele, okay, they give us small effect. They won't give us a lot of effects. I mean, that determines like blood group different. Why we say blood group different? Because blood group, you can see that it gives us direct effect. Okay, if you have IA, IO, definitely blood group A. So it means that it will give us the overall effect. But in this case, 180 genes, they interact together with different allele. They give us small effect. And this small effect is an additive effect. So it means that all of us will have a base. Let's say base 100 cm, for example. Oh, gene 1 add a little bit. Or gene 2 add a little bit. Gene 3 add a little bit. So it gives us a wide range of the variations. So therefore, if you measure, okay, if you measure the frequency distributions of the characteristic in the continuous variations always give us a normal distributions. When you have a large number of the sample, we always use histogram. Okay, or we use a normal distributions to represent our data. Okay, we're going to use the normal distributions to represent. So, okay, so the distribution curve that is frequency plots against the parameter. Okay, frequency plots against the parameter, you will get a bell-shaped normal distribution. A bell-shaped normal distribution. Are you clear? Okay, so you can see that these 180 genes interact together. You have to know. So polygenic. Each allele will give us a small effect, and this effect can be add on. So it means that it's additive effects here. So it gives us the final phenotype. Okay. So this is we have one question here that we can look at it. So continuous variations usually indicates the polygenic inheritance and additive effects of two or more genes on a single phenotypic characters. So height is a very good example. A recent study that involved at least 180 genes that affect the height. So we are not going to look at these 180 genes. We're going to look at the skin pigmentation. So how, what is the degree of skin pigmentation? How dark or how fair we are actually controlled by many separated inherited genes. Suppose that the skin pigmentation of organism is controlled by three unlinked. Unlinked basically means they are on different chromosomes. So we have E, F, and G, three genes. And each of them, they have what we call the big E, F allele or G allele. They contribute 2 times the units of the darkness. Okay, and you have the allele small e, small f, small g contribute 1 times of the darkness, right, to the skin pigmentation. So what does this mean? If someone with big E, okay, big F, big G, this person will have 2 plus 2 additive effect plus 2 times 2 times 2 times 2 times. So total is going to give us 12 times the darkness. So means that this is the highest, is the darkest, highest degree of the pigmentation. So if someone get the combinations of small e, small f, small g, then you're going to get 1, 1, 1, 1, 1, 1. So the additive effect will give us 6 times. So it means that this is the most lighter or most fair. Okay, this is the lowest degree of the pigmentation. So the rest of the phenotype will be in between of this. For example, so if someone with heterozygous for all, big E, big F, big G, small g, then you're going to get 2, 1, 2, 1, 2, 1. So the skin pigmentation will be 9, 4. Can I see that? So it means that in between. Okay, in between. So this is means by additive effect. Can you see that? So each allele gives us a little bit here and there to top up and make the final phenotype. Okay, so in this case, you can see that the homozygous here, big E, big F, big G, we have a 12 times the unit of darkness, very dark, or small e, small f, small g, it will give us 6 times with very light skin. So if we try with the parents, heterozygous parent, okay, so the answer, will be given later. You can see that 9-fold. When this 9-fold parents, okay, interbreeds or cross, then you're going to see that gamete genotype, we have 8 of them because they are unlinked genes. So unlinked genes mean independent assortment can take place. In the exam, they don't ask you to do this. This is just for practice only, okay? Don't worry about, oh, wow, I want to come at 8. 8 times 8, so means that's 64, okay? So you're going to see that this one, I already done everything for you guys already, okay? So you can see that different skin pigmentation, okay? Different skin pigmentation, you can see that different color tone here. So with the highest here, 12x, and the lightest 6x. So once you have done this, you can try to figure out, for example, if I start with 12x, I realize that only 1. Can I say that? If I 6x, only 1. But if you count 7x, so 1, 2, 3, 4, 5, 6. Can I see that? You have 6 of them. So once you quantify everything already, you will be able to plot. So what we plot, because it's a continuous variation, correct? It's a continuous variation. So by having 3 genes, I have how many phenotypes? I have 6 times, 7 times, 8 times, 9 times, 10 times, 11 times, 12 times. So I have 1, 2, 3, 4, 5, 6, 7 phenotypes already. Consider 3 genes. So if you have 180 genes, then you will have even more phenotype. So that's why when you plot the histograms later, okay, so given the graph here, you can plot a graph. So this graph is a histogram. So when you plot the histograms later, you will see that generally you're going to get the normal distributions. Are you clear? So what you plot? You plot frequency versus what? Versus the pigmentation, skin pigmentation. Then you plot a histogram, you'll be able to get what we call the shape, the bell-shaped curve. Okay. So now, to contradict or to see the different effects from the discontinuous, okay, then from continuous, then we're going to look at this discontinuous. So when we talk discontinuous variation, basically the difference is a qualitative. You only can tell the difference in color. You can see, oh yeah, it got different, but you cannot quantify that. So no way for us to quantify the difference. Cannot be quantified. Why? Let's say, for example, okay, we have person X and Y. Okay, person number 1, X, the blood group is A. The second person, blood group is B. What is the difference? So differences cannot be quantified. So when it cannot be quantified, this kind of variations fall into the discontinuous variation. Are you clear? Okay, so it's more to what we call the categorical. Categorical basically means that you categorize them based on their phenotype. Okay, so this is very clear into distinguishable category, or we call it as the categorical. For example, ABO blood group. So what is the main effect here? Okay, that what we have to know here. Highlight this one. So in discontinuous variations, a different allele at a single gene gives us a very large effect. And different genes have quite different effects onto the phenotypes. Let's say we got 2 genes. Gene number 1 code for blood group. Gene number 2 code for, let's say, blood group. Gene number 2 code for type of hemoglobin. Both of them involved in the red blood cells. But can you see that effect will be totally different? This one gives us blood group A, B, AB, or O. This one gives us whether normal polypeptide or abnormal polypeptide. So the effects will be totally different. And they don't have so-called additive effects and polygenic effects if compared to the continuous variations. Okay. For example, in this case, okay, ABO blood group distribution in Malaysia, you compare blood group A 30.50, 27.51, 7.50, 34.49, this kind of the data, what you're going to plot, you can't plot a histogram, okay, because it's a discontinuous variation. So we won't plot histogram. What we're going to plot here, we're going to plot a bar chart. Okay, we're going to plot a bar chart and it won't show us a normal distribution. Okay, you won't get normal distribution. So we're going to plot a y-axis of frequency again. Okay, and then versus blood group. Okay. So discontinuous variations does not show us a normal distribution. No. Okay, they don't produce a curve. We use the bar charts, okay, to represent our data. For example, in figure 15.3, you can see they use bar charts to show the blood group. Okay, so the last part for the first sessions here, okay, we're going to look at the differences between continuous variation and discontinuous variation. So in terms of definition, so continuous variation is a variation that has intermediates on the value that can occur within a population. So just now we say 180, 170, we can have 175. We can have 175.2, we can have 175.23. So that's why in this case, we cannot categorize them. So it's not categorical data. Discontinuous variation is variation that has a distinct group, categorical. okay, for organism to belong to, okay, blood group A, B, for example. Okay, what else? Let's say, for example, hair color, okay, or the eyes color, brown versus blue, okay, versus black. Okay, so example, the height, the weight, the heart rate, finger length, the leaf length, those are continuous variation. Discontinuous variation, whether your tongue can roll or not, your fingerprints, your eyes color, even though we have color intensity, but eye color is categorical, okay? You won't have a whole range of the color, okay? I mean, in terms of natural, and also blood group. Genetic basis, this is the most important one. Always remember, I summarize here. Okay, different allele and single gene locus have a small effect. And different genes will have the additive effect. And also we have the polygenics, means that we involve large number of genes that combine together, and we call this as a polygenics effects, okay? So polygenics, okay? So for discontinuous variation, different allele as single gene locus, they have a very large effect. And different genes will have quite different effect on the phenotypes, okay? So can we calculate the mean and average or not? Yes, continuous variations fluctuate around an average of a mean. So means that you're going to get the mean value here. For example, if you have whole population, you can quantify it. Okay, for example, okay, this person 170, another person 175, and so on. You can get the mean. But these continuous variations, you won't get the mean. Okay, for example, blood group A, blood group B, what's the mean? You can't get the mean in this case. So if an environmental factor, this one you can see that. Be careful when you use the word here. Environmental factor for continuous variations. Yes, environmental factor can change your or our variations. For example, body height. If you have, okay, for example, skin pigmentation, body height. Yes, again, the surrounding effects can have a very, very huge effects onto the phenotypes. But for this, okay, these continuous variations. Now, be careful, the word I use here is rarely, not means cannot, but it's very, very rare. Environmental factor can change our discontinuous variations. In terms of graphical representations, here we always use a histogram, or we use a normal distribution curve that show a smooth bell shape, okay? But when you use for the discontinuous variations, no curve is produced, and we use bar charts to represent the distributions. Okay, so with this, we have done the basis of this genetics, talking about the continuous variations and discontinuous variations. Okay, so with this, I stop the recording.